Welcome everybody to the second webinar of the online edition of the Protelec conference. So for the time being, the experiment is successful. Let's see whether this can go through also the second event. So the presenter of today is Huan-Xiang Zhou. I'm very pleased to have him here. So then the next week we will have Dr. Stephen Boxer. And then I would like to have your opinion, because from technical reasons we might need to switch from Wednesday to Thursday starting on February. So if nobody objects to this, and you can leave your comments on the chat if you want, we will switch the regular meetings from Wednesday to Thursday, starting on February. Okay, so coming back to our speaker of today, Dr. Huan-Xiang Zhou received his PhD from Drexel University in 1988. Then he worked for NIH, Hong Kong University of Science and Technology, again Drexel University and Florida State University. Then in 2017, he moved to University of Illinois at Chicago, where he is now professor in chemistry and physics. He's member of the American Association for the Advancement of Science and of the American Physical Society. His main activities on theoretical computation and experimental research on molecular and cellular biophysics. And he will be talking today about one of his interests, which is the binding kinetics of intrinsically disordered protein. Namely, the title of the presentation of today is actually regarding the role of electrostatics in the interaction behavior of disordered proteins. So, Huan-Xiang, you are very welcome, and you can proceed with your presentation.
Okay, so looks like I can share my screen. And okay, great. So I want to start by thanking Walter for hosting this series. It's too bad we can't visit him in Italy this time, but hopefully we can do that at a later time. I also just realized that I have a really tough competition, because this is the day that Joe Biden is inaugurating into a new president in the US. And so I'll try to make it worth your while. All right, so I'm going to talk about a subject that we've been studying for some time now, and I think we've really learned quite a bit even in the past couple years. So that's what I'd like to share with you. Before I start, I want to see. So I want to start by doing some advertisement and see. Can you actually hear me? I'm not sure I'm muted or not muted. Can you actually hear me?
Yes.
Okay. All right. So yeah, so I want to do a little bit of advertisement. So this is a review that I wrote a couple years ago now. So I basically put together, this took about more than 10 years in preparation. So I basically put in everything that I knew, and some of the things I even did not know too well, into this really giant review. I think was close to, must be, I even forgot now, I think close to 1,000 references cited. And so everything that as I said, I knew and did not know so well, I put together into this review. But I'm going to actually talk about more recent work. So mostly the talk is from 3 recent publications, actually all published last year. And so the first 2 topics about disorder proteins in solution and at membrane intersurface are two papers, one in biomolecules, the other is in Jack's Gold. And the third paper, if I get time, I'll talk a little bit about a very important protein that's involved in neurotransmitter release, this protein called synaptotogmin 1. I'll see what I get to that. So for a number of years now, my group has been very interested in various aspects of intrinsic disorder proteins. And one particular interest there is IDPs typically have more charged residues than folded proteins. And so therefore, we can anticipate a lot of electrostatic effects, as I will highlight.
Huan-Xiang, sorry to interrupt you. Can you share, you are sharing the presenter screen and not the presentation one. So we see your personal screen, not the presentation one. So you may need to change the screen you are using.
Okay, yeah, hold on, let me do the full, okay. How's that?
Yeah, okay. Much better. Thank you.
Okay, so yeah, so as I said, so we're interested in both practical problems as well as sort of more fundamental issues, right? Because there are a lot of really basic questions that are raised by structural disorder, right? So in the old days, we all learned from biochemistry books about the central dogma of molecular biology, from DNA to RNA to protein. And we also, I think, corresponding, there is what I would call a central dogma of protein biophysics, where you go from sequence to structure to function, right? But when you have proteins that don't have defined structures, then this link then breaks. So in particular for disorder proteins, how does the sequence code for conformation? So no, we don't have a unique structure, but we still have a conformational space, right? And the space may not be uniform. So what codes for the landscape in the conformational space, as well as protein dynamics and protein function, right? So what I hope to do today is to give you some hints of what we've learned in addressing this various questions. So this one set of disorder proteins that we've been studying, or rather proteins with disorder regions, come from proteins that are involved in cell division in the bacterium that causes tuberculosis. And those are the proteins that you form this complex called the diffusome, which is the machinery for involved in dividing MTB cells. And of course, this is a process is crucial for pathogenesis, as well as for antibiotic resistance. One thing that's worth noting is that the inner membrane, so mycobacteria are, they have obviously two, like in most bacteria, they have two membranes. So the inner membrane is highly enriched in negatively charged lipids. So these are the PG lipids, cardolipin, and they account for more than 70% of the composition, right? So you wonder, what are all these negative charges doing? So one thing that struck us right away is if we look at the number of proteins that are part of the diffusome, right? That Chi-Z, CRGA, these are all bacteria. People then actually use four-letter terms for their proteins, so Chi-Z, CRGA, FTX2. So they all have disorder regions. So the green sequences form transmembrane helices. So what's before that is in orange are the cytoplasmic regions. And so in all these three cases, and as well as in others, the cytoplasmic regions are disordered, right? Now, if I color code the positive charge and negative charge residue, you'll see right away there are a lot of positive charges, not for arginines in particular. And so that's true in Chi-Z that I'll be talking about in a moment. But there are, and that's true also in a number of other proteins, CRGA, that we're also studying FTSQ. FTSQ is also special in that there is a charge segregation. So the first half you see a lot of E's and a few D's, and the second have a lot of R's, right? So again, highly concentrated in charged residues, right? So that raises again interesting questions for us, right? So first, we're going to study the just the disorder region of Chi-Z, the first 64 residues that's preceding the transmembrane helix in the cytoplasmic region, right? So I already talked about the enrichment of positive charges, and in fact, if we look into the sequence even bit further, we can even further divide the 64 residues into two halves that we call N half and the C half, but first, I mean, there are now, as many of you know, there are a number of disorder predictors, and if we put in those predictors, we see it's highly almost all these predictors predict almost 100% disorder, and we can easily check disorder by very simple NMR experiment. That's the HSQC spectrum, right? So disorder proteins have a very simple character, and that is they have very narrow dispersion in the proton dimension. So proton chemistries are very similar among different residues because disorder proteins, they have very similar chemical environments. So therefore, there's not much dispersion in the proton dimension, right? So as in this case, as well as in many other cases, this HSQC spectrum right away tell us that indeed this Chi-Z N-terminal region or N-terminal domain is highly disordered, and so a lot of positive charges, but even the positive charges are distributed differently according to the N half and the C half. So there are three negative charges. They're all in the first half or the N half, right? And so the net charge in N half is +2, but the net charge in the C half is +8, right? So we see this asymmetry. And another asymmetry is the distribution of pronins versus glycines, right? So pronins and glycines are special because they break secondary structures, right? So pronins are propionated. Pronin is also special for forming polypronin 2, as I'll point out that's important, right? So pronins are highly enriched in N half. Okay, 2/3 are in N half, but then in contrast, glycines are mostly located in the C half, right? So you have this asymmetry again, and so that's going to affect conformation and the dynamics, as I will show in a moment. So now, so we do this by NMR. We'll also do this by all atom explicit solvent molecular dynamic simulations. Now, traditionally, the force fields have been parameterized for structured proteins, and we apply those to disorder proteins. It has been found out that they need to very overly compact disorder proteins. So therefore, the first thing we need to do was to actually select a force field that's appropriate for disorder proteins. So we tested a number of different force fields, and against sex data, against NMR data. The long and the short is that we found one particular force field. This is the combination of Amber 14SB with a water model that's from the D short group, TIP4PD, and so that's the combination force field that we're going to use, right? So we did very extensive simulations. These are 20 some trajectories. Each is multiple microseconds. So we basically simulated the system to deaths on the GPU cluster, and consistent with the prediction of disorder and NMR verification, there is very little second structure in the MD simulated confirmation, but there is a high population of polypronin 2, as indicated by the blue bars, right? And also there are several regions where the PP2 propensity is particularly high. For example, around residue 28, around the residue 12, and those are all regions where you have pronin residues in the midst. And looking at the conformational space as from a global point of view, really it's very, very flat. There is really no well sampled conformations, right? So this is a set of 16 that we choose as a representative, but really nothing can really represent this conformational space. However, there are other features that are recurrent, okay? And that's in particular salt bridge. So we see a lot of salt bridges. We see a lot of again, consistent with the high PP2 propensities, we see the PP2 regions as indicated by the purple backbone, and very few, if any, helices and beta strands, right? And so does that mean there is this protein is totally disordered? There's no any kind of feature? The answer is no. And so we actually do further analysis by calculating context, right? So we look at the side chain context, side chain backbone context. Now we see that there are actually local segments that emerge that are characterized by side chain interactions. So for example, so basically they separate, there are 5 different segments. It's a little bit overlapping sometimes. But if we look at, for example, box 3, which is a stretch of residues from roughly from 12 to about 30, right? And so we see a lot of side chain interactions, right? And so these are illustrated by this particular conformation where we see salt bridges between arginines and asp, and we'll also see cation pi interactions. And so now remember that all the 3 negative charges are in the N-terminal half. So therefore, salt bridges are populated in the N half. But sometimes they do form, they reach out, this negative charge residue do reach over to the C half and also form sort of more distant salt bridges. But overall, it's the N half that's more, that has more side chain interactions, right? And so that's going to impact conformational dynamics, right? That we now turn. So we've studied the backbone dynamics by NMR. So NMR is really good at characterizing timescales, dynamics on different timescales. In this set of experiments, we measured dynamics, backbone dynamics on the timescale from picoseconds to a few nanoseconds, as reported by 3 different properties. These are the longitudinal and transverse relaxation rates, as well as nuclear Overhauser enhancement that are measured for individual residues. Just to orient the audience, so R2 is mainly influenced by dynamics on the time scale of a few nanoseconds, right? Whereas R1 is most influenced by sub-nanosecond, a few 100 picoseconds to maybe 1 or 2 nanoseconds. Whereas NOE is most influenced by dynamics on the tens to hundreds picosecond time scale. They really report time scales on different dynamics on different time scales. Okay, so so ranging from short for NOE to intermediate for R1 to long for R2, right? And we see that especially for R2, right, that it's not the R2 rates are not uniform. We see 3 regions that show local maxima, and that correlates directly with the interactions that we have identified, right? So the residue, so there was a peak around residue 26, right? That's directly correlated with the subspace in box 3, and then we see the peak around the residue 47 corresponding to a subspace in box 5, and then there is also a local minima that correspond to actually 2 glycines, residue 41, 42, and actually, so there are actually 2 traces. The black traces are experiments, right? The calendar traces are actually are predictions from the molecular dynamics simulations. So we do get very quantitative agreement. So remember, this force field was the parameters based on largely based on conformational properties, NMR chemical shifts and sex profiles. But here we actually see the same force field actually also predicts well dynamic properties. Really shows, I think this combination really, we've now actually expanded this to a dozen other IDPs. They all do very well. And so I think we this force field is really very well parameterized for simulating IDPs. And so the MD prediction is based on calculating the energy bound vector time coordination functions. And so from these time coordination functions, then we typically fit them to a sum of 3 exponentials, and then we do Fourier transform to get the spectral density, and then from the spectral density, this J omega. From this, then we can predict R1, R2 NOEs, right? So there are residues that have very big components in the slow time scale, so what are the 3 time constants? Tau 1, tau 2, tau 3. So tau 1 is in the 5 to 10 nanosecond time scale, tau 2 is in the 1 to 2 nanosecond time scale, and then tau 3 in the sub nanosecond. They really, each is make the most important contribution to one of the 3 NMR properties, right? So tau 1 contributes the most to R2, tau 2 to R1, and then tau 3 to NOE. So now look at the protein as a whole. We see that amplitude for tau 1, that's in this magenta color, right? That has a higher than average. So basically, you can see the protein breaks into N half and C half, right? So the N half has a much higher tau 1 amplitude than the C half, and that again is related to the fact that you have many more tertiary and interaction solid bridge hydrogen bonds, and as well as polyprotein 2 propensities, right? So all these structural features, they translate into high amplitudes for the slow time scale dynamics, and so we have asymmetry between the 2 halves, essentially, right? And another way to characterize this background dynamics is by amide proton exchange, where we basically measure the extent to which backbone amides are protected from exchange with solvent, right? So the higher protection means slower dynamics, right? And here again, we see that the C half, the protection factor is very close to 1, meaning that it's really not much different from model peptides, whereas the N half is about 2 to 3-fold protected relative to model compounds. So again, indicating the presence of those interactions. So overall, I think we conclude that because of interactions, tertiary interaction, solid bridges, cation pi interactions, as well as formation of polyprotein 2, we find there are correlated segments that drive large amplitudes on the slow time scale, and there's also a symmetry between the 2 halves, and so we think that this kind of asymmetry, or the correlated segments, may be vehicles by which IDPs really code for local conformation and backbone dynamics, and may even code for how they function, as we'll turn to next, right? Now this is NT, or the first 64 residue, is part of a full-length protein. Again, that has a transmembrane helix, as well as on the paraplasmic side, there is also a structured domain, right? So that's the Phoenix Chi-Z protein, right? And now, given the high percentage of positive residues and the highly negative nature of the MTP membrane, so we anticipate a significant membrane interaction. So that's what we now turn to. So again, to motivate our computer study, we did NMR experiments to confirm that indeed there is membrane association. So again, here we again use the same NMR technique. This is to measure HSQC's spectra, right? So we now in this case, the experiment is done in the presence of liposomes. So we form liposomes using different lipids, POPC, which is a right ionic or neutral lipid, and we see that there is no membrane binding. So that's an easy way to indicate whether or not there is membrane binding is look at whether or not you have all the peaks present, right? If all the peaks in the solution are present, so the solution spectrum are shown in black, and then the spectra in the presence of liposomes are shown in red. So if all the red peaks have the same, the red spectra have the same number of peaks as the black ones, then that means there is no membrane interaction because the protein, the liposomes are not doing anything. So so the spectrum is intact. So that's true of POPC and also POPC DOPE mixture, and that's also neutral. And then panel C is where we actually now start to mix POPC with POPG, which is actually negatively charged, but at a 20% level, 20% negatively charged. We see that that's still not binding, no membrane. But when we get to 70% of POPG, now we see most of the red peaks disappear, a few that remain. So that's now indicating membrane association, right? So now the ones that are remaining are also interesting. So all those remaining peaks are near the two termini, right? With residues that are near the extreme C terminus, like arginine 62. We also have residues, peaks that are near what appears to be the N-terminal residues. Here we cannot really make full assignments, but they do suggest that the termini are still free, and there is some interaction in the middle, right? That's all as much as we can tell from the HSQC spectra. But we have other, NMR is great because there are many different kinds of experiments that each tell a different story, and so here we turn to a different set of NMR experiments. There are actually two kinds of experiments here. One is called INEPT. So INEPT is a technique that detects dynamic residues. So if the residues are very floppy, then they show up. There is also an opposite experiment called cross polarization, or CP. CP detects static residues, right? So by combining these experiments, then we can get a pretty good idea what residues are static, what residues are dynamic. And so based on two things, I can summarize. Most of the residues are dynamic, right? So the black cross peaks are basically contain almost all residues, right? So that means that even though Chi-Z is bound to the membrane, but it remains highly dynamic. Hence, we'll call this association fuzzy. But there are a few peaks that are that show up in the CP spectrum. These are most notably arginines, right? And pronines, alanines, and maybe one or two leucines, right? And so I'll for sake of time, I'll focus on the arginine residues, right? So the arginines are basically, this says the arginines become more static, right? And that's really indicating that they're involved in membrane interaction. And when I will confirm that using yet another NMR technique called PRE, or paramagnetic resonance enhancement, right? This actually directly detects membrane interaction. So we actually dope the membrane with a indicator that actually lets you tell whether or not there is interaction, right? So so we actually have a spin label in the membrane that tells you that there is interaction, right? So we see that that the arginine peaks in the presence of the spin label are suppressed. Okay, that's because of the resonance enhancement, right? So now there is because cross polarization, so now the peaks are reduced. So and that's true largely of arginine residues and not so much for other residues, right? So this confirms that it's arginines that are interacting, right? So that's the story from the experimental side, and then we did again lots of 16 trajectories for and again multiple microsecond each, right? So this is one simulation of the just the N-terminal region by itself. It's very dynamic, as we've indicated, but again, there are arginines that really that's holding the protein together with the membrane, right? It's not the same arginines. Sometimes it's this arginine, sometimes there's other arginines, but they're always a subset of arginines that are stabilizing the interactions, and hence explaining the dynamics as well as the prominence of arginine in stabilizing this binding process, right? And let me now, yeah, I think I want to actually move on. So we can tally up the statistics, right? We find that indeed arginines form hydrogen bonds. I should point out we actually studied 3 different systems, right? So Chi-Z, as indicated by the number of residues, the first 64 or the first 86, that also includes the transmembrane helix as well as the full-length protein, right? So the N-terminal region, the interactions are largely similar across the 3 different constructs. There are subtleties, right? That near the especially near the N-terminus where it connects with the transmembrane helix, right? So obviously the ones with the helix are going to have the N-terminal residue, the C-terminal residues rather are going to have a high propensity for membrane interaction because they are tethered by the transmembrane helix to the membrane, right? But here again we see now asymmetry, right? Earlier I talked about in the unbound Chi-Z we see asymmetry involving intra-protein interactions that are populated in the N-half. But now we see membrane interaction has the flipped symmetry. Membrane interaction is favored in the C-half, right? So really there is a competition between intramolecular hydrogen bonding and intermolecular interactions, right? So whatever, when you are involved in intraprotein interaction as in the C-half, then you lose the ability to interact with the membrane, right? So here's another movie showing this asymmetry, right? You see that the N-half is largely free, or the C-half is most of the time is bound. Okay? And that asymmetry is accentuated by membrane tethering, right? Because the C-half is right just preceding the transmembrane helix, right? So all right, and so to conclude this part, I think I've shown evidence indicating that Chi-Z association with membrane is fuzzy. In fact, it's extremely fuzzy. There's no gain in segment structure. It's highly dynamic, but that does not mean it's random. There are actually important characteristics, and the most important characteristic is that the positive charged amino acids are really what's driving the interaction. It's really the overriding sequence requirement, right? So I think from this and many other studies, I think the important lesson that for us is now we can actually now define general rules for sequence requirements for membrane association. So we're actually now developing actually a predictor for a membrane association. And of course, membrane association of disorder proteins is implicated in many cellular functions. So actually Walter, maybe I can take a few more minutes to tell the cinematography story, or
sure, yeah, absolutely.
Okay, so yeah, so thanks. So this is a protein that actually is highly important. It's actually involved in membrane fusion, specifically in the release of neurotransmitters. Most of you probably have heard of the SNAC complex that serves as the fusion machine. But the trigger for this process is the synaptotogmin 1, because this process is triggered by calcium. All right, so synaptotogmin is really the sensor, the calcium sensor, right? So once it senses an influx of calcium, then it initiates a series of processes that allows the SNAC complex to do its job, right? So now what the SNAC complex does is now very well characterized. What is still controversial is how this process is triggered by synaptotogmin 1. Okay? So that's what we try to learn something about, right? So so synaptotogmin 1 is indicated by this sort of lollipop type of illustration, right? It has a single pass transmembrane helix. So it's residing in the vesicle membrane, and it has two structured domains that binds calcium, hence the calcium sensing ability. And so ultimately, it leads to the fusion of the vesicle membrane with the plasma membrane, and it results in the release of neurotransmitter, right? So that's the biological context, right? So that's the synaptotogmin protein sequence and structure, right? So again, we have one single transmembrane helix followed by a 60 or so residue disordered linker that again is a highly enriched in charge residue. There is also charge asymmetry. All right, so the first portion of this linker is highly enriched in positive charge residues, and then the ending segments is highly enriched in negative charge residues, right? And then followed by two what's called C2 domains. So C2 domains bind calcium. So specifically the first C2A binds 3 calcium ions. The C2B domain binds 2 calcium ions. They are structurally similar, and in terms of sequence, they're also homologous, right? So that's the system that we're studying. We are again studying a number of different constructs, ranging from cutting basically different pieces off, right? So we have a system where we cut both of the C2 domains off, and then we have a system that cuts only the C2B off, as well as the full-length protein. And we study charged. So the plasma membrane, the inner leaflet is also highly enriched in negative charged lipids, especially PIP2. So PIP2 is a highly negative charge. It actually has a net charge of -4, and then phosphatidylserine or PS has a single negative charge. So we then studied the interactions of synaptotogmin 1 with discharged lipids, right? And so we first look at the linker. How does the linker, would the linker interacts with membrane, given that we know in other systems, the linker actually interacts with membrane. So in this case, actually the linker has minimal interaction. And in the only near the N-terminal region where you have those basic residues where you have some interaction, the rest of the linker is really not interacting with the linker at all. And so this is illustration of that. Let me see whether doesn't look like this movie is going to play. All right, never mind. Okay, so moving on. So we look at then what's interacting with the membrane are the two C2 domains, right? So the C2 domains, again, each has a calcium binding site, and the calcium binding site is what's interacting with, in this case, is the calcium that's actually mediating membrane interaction, because the binding site has not connective charges. Without the calcium, they have this region actually does not have strong calcium binding abilities, but upon calcium binding, then this loop becomes high propensity site for membrane association, right? So both C2 domains associated with membrane, but just like in the Chi-Z IDP case, even in this the case of these two structure domains, the binding is not stable. There are actually different poses, and so it actually samples the C2 domains actually sample a range of poses, and with different tilt angles and involving different residues, so here again there is a dynamics in the bond state, and the membrane interaction when becomes more stable when you have PIP lipids. Now I should have pointed out the vascular membrane is only PS, whereas the plasma membrane is PIP, right? So you have this more negative charge lipids in the plasma membrane, but not in the vascular membrane. So so the membrane association becomes much more stable and less dynamic in the presence of PIP2, right? So that's one difference between the vascular membrane and the plasma membrane. Now we also found that so that means that vascular binding is not so stable. And whereas the plasma binding is very stable. So initially, when the process starts, right, the synaptogamy protein is bound to the vascular membrane. So the C2 domains are initially bound to the vascular membrane, and that binding is unstable, and there is in this case there is also competition between intermolecular binding, and the intramolecular binding is between the linker, the negative charged portion of the linker, and the C2b, right? So in this case, this competition again pulls C2b away from the the vascular membrane, and hence turns over the C2b domain, right? So let me see. Okay, here we actually can play the movie. So this competition leads to the turning over, right? So now the calcium binding site is pointing not towards the vascular membrane but towards the plasma membrane, therefore priming it for binding with the plasma membrane, right? So once it binds to the plasma membrane, then it becomes very stable, right? So now we have this result in this sort of a breached version where we have the protein that's connecting the two membranes, right? Bring the two membranes together, and I think that is then. So let me see what I can. Play a short movie, right? And so it turns over, and then binds to the plasma membrane. And so to summarize this thing in a simple cartoon, then what we think how synaptogamy 1 triggers the fusion process is through membrane binding and a dynamic. So initially, the two C2 domains are bound to the vascular membrane, and then through the competition with the linker region, then the C2b flips over, and then that lets it bind the plasma membrane, and then so that you have this bridged configuration. Then once this bridged version is formed, then C2B has an additional role in serving as a platform for the full assembly of the snare complex, which then finally leads to the fusion of the two membranes, the release of the vesicle. Right. So with that, I want to acknowledge the people who did the work. So the Chi-Z computational work is largely done by Anna Hicks, and our experiments is actually done in my collaborator Tim Cross's lab, and his student Christian Esquire is the one that actually did all the experiments. And then the stegatomic work is done by a postdoc from Mesh Prasad in my group. Thank you very much for your attention. I'd be happy to answer any questions.
Thank you very much for your presentation. So before heading to the questions, so I would like to clarify two things. One thing is that the next meeting will still be on Wednesday, is confirmed as the program is saying. And for the switch to the next month, I will need, since I got some comments on the new day, I will need a bit more time to find a time that suits everybody, hopefully, or almost everybody. So for the month of February, we are still a little bit on hold. So, okay, now we can go over with questions.
So I have a question. Thank you for the interesting talk. I actually have a couple of questions. One has to do with in the beginning you showed various conformations for the IDP. So for that slide, I'm just curious. Yeah, you showed how flat the landscape is, and yes, this slide. So so what's the sort of in terms of KT? What's the difference in KT between these conformations? Is it a fraction of KT? Is it around KT? So can I get a sense of how far they are?
Scale bar. Let me see whether. Hi, Alex.
Yes. Hi.
Hi. So we have a scale bar. So I don't know.
Yeah, I'm just trying to say, you know, with this, is that fraction of a KT? Is that?
Yeah, it's probably a couple of, right? So the before the yellow, basically, it's a couple of KT, right? So the blue and the light blue, they're all within
the light blue. Okay.
Yeah.
So there's a couple of KT. I see.
Yeah.
So it's not a fraction. Okay. All right. Thank you. So my second question is more of a, I don't know if you've thought about it, more of on the methodological side. The tip for PD water is its dielectric constant is about 15% off experiment. It's lower than experimental by about 15%. Yet, as you've shown, it works pretty well. So I wonder if you thought about it, you know, why is that, especially for a highly charged peptide like this, is that because the force field kind of overcompensates? Or is that really, you know, the difference between like 65 and 78 doesn't really matter because it's 1 over epsilon?
I think it's probably a little bit of both. We're mostly interested in, or our studies has been focused on protein-water interaction, and so that part appears to the pure water property may be somewhat slightly off, but the protein-water interaction seems to be pretty well modeled. We actually have another study where we look at rotational dynamics, and we find that that's also, yeah, it's for structured proteins. It's also works not only for disorder protein, but also for structured protein. Actually, does a pretty good job in predicting rotational coordination times. And so it looks like it's so maybe, yeah, there is a balance, right, that somehow is achieving the proper balance between, maybe if to the extent that the dielectric constant is smaller, so that there is it's not screening, for example, I guess electrostatic interaction is strongly, but maybe there is some kind of compensation that's going on that is achieving the right effect. But certainly, I think you do read. I mean, it's interesting questions about whether, I mean, this under prediction of dietary constant, whether or not that translates into something, yeah, to keep in mind. Yeah.
Thank you.
Okay, so I have a question from Anna Damjanovic.
Yeah, hi. So I also have a couple of questions. So the first one is, so very interesting talk. Yeah. So
Thank you. Are you in Baltimore?
Yes.
Okay.
So so the first one is, so for the first part of your talk, did you have lysins in there, and you probably didn't see interactions with lysins with the membrane, because so you were mentioning
there's no lysine.
Oh, there's no lysine. Okay, so
this particular protein, is all arginines. There are other proteins. There are other proteins that we've studied that do have lysines, but not this one.
Okay, do you see lysine interaction with the membrane or no?
Not as much. In the cases where we have both arginine and lysines, arginines always does much better than lysine.
Yeah.
Yeah.
Yeah. One would think so.
Yeah.
And then the other questions are for the second part. So how much experimental structural information you have, and when you did the simulations, what were the starting structures? Because you see like nice binding to the membrane with the calcium ions. So do you start it from that conformation, or you start from other conformation?
The snaptocomine. There is no, so we have crystal structures for the C2 domains. So that's the structure that we have. And so initially what we did was we placed the C2 domains near the membrane, but not directly interacting with the membrane. And then they just, during the equilibration, they simply pick up the interaction, the membrane interaction.
Okay. And the other one is also, did you look with what does the structure look like without calcium, right? Do you know about it? Because you would have then probably some cluster of many carboxylic residues, right?
Yes, yeah. So that's right. So without calcium, the certainly the calcium binding loop does not bind with membrane. There is another region that some people have proposed to be. There is a region called polybasic, the stretch of lysines on the side that have been proposed to be important for binding PIP-containing membranes. That we do show that's involved in membrane association in conjunction with the calcium binding loops. I'm not sure on their own they can stably bind with the membranes, even if they contain PIP.
Okay.
Okay, then there is a question from Franz Mulder.
Yes, hello. Thanks for a very nice talk, and congratulations on the inauguration. It has successfully been done.
All right.
for Marilyn and others over there as well. I had a question about a few things. So you reported in the beginning that you see a whole lot of polyproline 2, and I was wondering if this is something that you extract from the simulations just on the basis of what the force field sampling is. Is that correctly understood?
Yeah, that's right. It's based on the simulations. So there is no simple way to detect, initially in the NMR experiments that we carried out, to detect PP2.
So there are a couple of ways to detect polyproline 2, right? The 3-bond coupling experiments, but you haven't. Yeah,
there are not the kinds of experiments that we did.
Okay. Yeah. Because that seems quite a bit higher than reported for most IDPs or IDRs, right? So it's quite
that's partly because the high protein proportion, right? I think it's So it's 8 out of 32. So that's every fourth residue is a proline in NHAF.
Right, right.
Yeah. That is also higher than some of other IDPs that were simulated. Yeah, that it contains less proline residues. Yeah, that is a bit unusual in that sense. Yes.
And a question about and the same thing actually for the salt bridges. So is it really is it possible in some way to validate that as well? I mean, how does it work? For example, if in your simulations, if you can you screen charges in a similar way as you could do an experiment, and then you get also the similar response of your system?
I suppose one way to do that it would be to see how backbone dynamics is affected by adding salt, right? That would be one way to validate, because our prediction is that this higher than average R2 values are partly resulting from this kind of salt bridges. So the guess would be that if we screen out the chance of forming the salt bridges, then the backbone dynamics will become more uniform across sequence.
Yeah, because some other investigators like Marcus Zweckstetter, for example, they reported that the bulkiness of the side chain, so it's more like an inertial effect, it seems, that seems to restrict the amount of backbone motion that you have, right? I don't know if
I personally don't think that's likely, because we have arginines only because we have so many arginines, but it's only specific sites that have higher than average values, R2 values. So I don't think the bulkiness may have something to do, for example, with amide proton exchange. That may play a role, but I think in terms of NH bond relaxation, I don't think the bulkiness is, at least from my reading of the NMR relaxation, has anything to do with the bulkiness.
Yeah, there's some older work that shows a very clear correlation between these two things. So bulkiness and the backbone order parameter. Yeah. So the R2 especially. The local correlation time of the NH bond vector.
Yeah. Can you actually? I'm interested in this. Do you mind drop me a message?
No, I can do that. Sure.
Yeah, thanks. I'd like to actually read about it. Even though again right now, by comparing arginines at different sites, we don't see that as a big effect.
Okay.
But maybe comparing arginines as a whole versus, I don't know, alanine as a whole, maybe that might be some difference that show up. I was just comparing arginines from different sites, and there was unevenness among arginines. Yeah, that was our focus.
Yeah, it's kind of hard to probe experimentally. I agree.
Okay, so I have a question from the chat from Tom Lauer, who is asking, how are solvent ions handled in your models?
Could you say it again?
How are solvent ions handled in your models?
Oh, this we don't actually do anything special. This I think we simply put in the nominal 150 millimolar salt. There's nothing special done on the salt.
Okay. Then there was a Mary who wanted to ask something.
I'm sorry?
For some reason, my hand won't go up, and so very nice. So one question is, like, what is the dependence on the lipid model? So since you're looking at this arginine probably phosphate interaction, how sensitive is that to what lipids you're using?
Yeah, well, so in that we're basically limited of what lipid models are available. Let me go to where are the thought I had this lipid. Yeah, right. Yeah, so these are we're basically using what's available in Amber, and so that's the model that we did do a little bit of comparison of different especially for PIP2, different PIP2 models, and the comparison we made we did not see any kind of quantitative difference. Yeah, so we checked with lipid 17. This is in some of our earlier simulation. Then our recent simulations is actually using the lipid parameters in amber, and those two, they don't seem to have much of a difference.
I was just thinking about the net charge, whether it's Witter ionic or charged or.
Oh, the charge, yeah, of course, that's going to be a big effect, right? I mean, it's, yeah, obviously, that's, yeah. So that can be tuned obviously by pH, right? I mean, that if I think if you go to really low pH, then yes, some of the phosphates will be neutralized. I mean, I think that will entail you go to really very low, like pH 4 or pH 3, something like that, and
also, if you change the lipid type, you'll see the charge change.
Yeah. So yeah, that's right.
Rafts forming around the arginines. I mean, you have lots of you were very protein centric here rather than.
Well, the lipids do matter because I mean that's why PIP2 is so special, right? That There are this phosphorylated PIs. I mean, PIP2 is there are many different species of phosphorylated PIs, right? You have PIP1, PIP2. You can even have PIP3, and they do make a huge difference.
But their concentration is relatively low, right? So you don't have a lot of those.
So there are actually two things to say about, even though on average, right, is 4%, let's say, there are two things I think that really sort of skews the picture. One is the lipid composition is asymmetric, right? So PIP molecules are almost exclusively on the inner membrane. That's one. So that immediately raises the composition from 4% to 8%. Number two, there is now, I think, strong experimental evidence to indicate that even within the inner leaflet, the distribution is not uniform across different regions. In fact, at the fusion sites, it's been shown that PIP2 concentration is much higher than average. So the local
that's interesting. Do you know what's causing making the concentration higher at the fusion?
I guess it's sort of chicken egg question, right? And it's so there may be right this proteins, say synaptocogmin, other proteins, right? They like to interact with the PIP, right? So therefore there is this natural partnership. There may be, so this sort of all passive, there may be, I think, more active components that's involved too. But that, I think it's still unknown. But what is known is that the local concentrate compositions are adjusted. And so the local may be much higher than, so when we read literature about, I mean, average lipid levels, I mean, that's really not telling the whole story.
Okay, I have one more question from Tom Lauer, who says, experiments demonstrate that PEs are anionic and not neutral.
Okay. Ionic, I mean, it's right ionic, so I don't know what that's what it means. Neutral is only in terms of net charge, right? You have this coning group that's positive, and then phosphate is negative. So the net charge is 0, but there are two charges, one positive, one negative.
Probably he meant that it should be globally negative, anionic, when he says anionic.
I'm not sure how that, it's just chemically how that's And I mean, of course, unless you go to high pH, right? That's when you neutralize the amide. That may be, but I think at a neutral pH, then it's likely that a Koenig group will be positively charged.
The experiments demonstrate that phosphatidylethanolamine is anionic. It's almost as anionic as phosphatidylserine in neutral pH. I'll send you the paper. It's biophysical
Okay.
journal.
Okay, all right.
The other thing is that when you start getting to high concentrations of some of these phospholipids, they start exhibiting polyelectrolyte behavior.
Ah, okay, okay. So,
And that's going to be true on some of your peptides as well. Some of those stretches of amino acids, you're below the Gerum length. It's going to start exhibiting polyelectrolyte effects, and those will be interacting now with the solvent ions. That's why the question to my earlier question about the solvent ions. In any case, thank you. It's a wonderful talk.
okay, all right, thank you. Yeah, so but please do send me the paper that you were referring to.
I'll be glad to.
Thanks.
Okay, I have a question from Antonio.
Okay.
Okay. Well, first of all, thank you for this really nice presentation. When you show, I'm going back again to the energy landscape that Alexei was commenting before. Is that a principal component analysis?
Yes, yes. This is I think torsion-based PCA. Yes.
Mm-hmm. Yeah. Well, my question is more of a comment, maybe, because, in fact, for such a highly disorganized system, where you have such large portions non-structured, I would expect that the first modes in a principal component analysis, which I suppose are the ones represented here, right? Because you have mode 1 and mode 2 there. I would expect those ones, since the first principal components, the criterion is simply to try to find the directions along which the overall dispersion is the highest one. Then I would expect that the local structure may be hidden in higher order modes. So in some of these cases, may be helpful to look even at simple histograms of the other modes, of mode 3, 4, 5, because you may have structure in the conformational space, but the local minima may be all lumped together in this big central well.
So I should point out that I should point out that the eigenvalues are very smooth. I mean, there is, so the first two modes are not, I mean, basically it's not, the eigenvalues are very close to each other. So we're plotting the first two only for convenience to make the point that it's flat.
So it's flat on the higher modes as well.
Yeah. Yeah.
Okay, that's interesting.
There's nothing that's standing out. Yeah.
Okay, okay. Yeah. Because I have a similar problem with a different kind of system, which were peptidic dendrimers, and in those cases the system was essentially uniform in terms of composition. So I was expecting that in this case, maybe hopefully, since your system is not homogeneous as my dendrimers were, that you could see something in the higher order modes, but apparently you can't. Okay.
No, yeah, it's the spectrum eigenvalues is very gradually going all the way to the end. Yeah.
Okay. Yeah, bad luck then. Okay, thank you.
Sure.
Okay, it looks like there are no more questions. Okay, no, there is one comment from Stefan Herbo on the chat, who's saying, a follow-up to Antonio's question. What is the variance of capture, and potentially would it be more advantageous to apply time lag independent component analysis?
Again, I think this is this whole analysis is only to make a very trivial point, right? That the antigenicity gap is very flat, very featureless. So therefore, we did not feel it's justified to do any kind of further analysis. Now, if there were sort of interesting features, I would have pursued. But really, this is I only presented it as a sort of null result, and so that's why we did not spend too much time on doing any kind of detailed analysis.